Thank you. I'll be talking very generally, actually, instead of on a specific system, or I'll be talking more about chromatin and different cells, and more about technology that we've been developing to hopefully solve problems that many of you have or would like to study. So in general context, we think a lot about what's actually in a nucleus. Here, where chromatin in all eukaryotes, or almost all eukaryotes, is packaged into nucleosomes, which are histones wrapping DNA. And what's important for the function of chromatin is really where are specific chromatin components? Where are these nucleosomes? Where are transcription factors that bind DNA between these nucleosomes? Okay. So the work I'll be telling you about today is really work that I've been collaborating, I have it right here, collaborating with Steve Henikoff's group at Fred Hutch. And the work is really of 4 different postdocs, Derek, Jay, Hadiye, and Sandipan. And I'll be telling you really 3 different vignettes of work we've been doing. And the general problem, really, is that we'd like to be talking about different cells and what their chromatin looks like in different cells in different aspects. And so you're familiar with the idea that every kind of cell that we look at will have differences in gene expression that determines what those cells are and what they do. And we have ways to measure gene expression, and typically you can think about measuring mRNA in cells. But I want to emphasize that an important aspect of the differences between cells is not only what they're expressing, but what they're not expressing and actually intentionally silencing. So you can think of these 3 imaginary cells here that, yes, they express a certain set of genes, but each of them also deliberately silence another battery of genes, which is what distinguishes 2 different cell types from one another, what they express and what cells from the other cell type they're repressing. Okay? And we can look at this in terms of chromatin features of those regions for typically for active histone modifications like histone modifications of lysine 4 dimethylation are a good marker for what regions of a genome are active. And lysine 27 trimethylations are a very good marker for what regions are silenced. So we can think about looking at both of those components to distinguish cells. The normal way that you would look at chromatin in any cell is really by chromatin immunoprecipitation. And we had a lot of trouble with this technique. And the reason is because actually, even though it's a very commonly used method, it has some fundamental problems which really limit what you can do with it. And this is a diagram just to sort of emphasize what those 2 problems are. The method in general is that you take a cell you're interested in and you immediately fragment all the chromatin in that cell into many, many small pieces. Okay? There's different ways of doing this, but the point is that now the whole genome is broken up into pieces. You have to sort of delicately handle that in a test tube, and there's a lot of different pieces. And as a second step, if you want to find where a specific transcription factor is or where a specific histone modification is, you use antibodies to that component of interest, mix it in with your fragmented genome, and try to immunoprecipitate the piece that you care about. And the problem is there's a lot of different pieces, and really you're relying on the efficiency, the specific affinity of your antibody to recover just that piece out from the vast amount of other non-specific pieces. Okay? Now, and that's really the problem that limits the effectiveness of this method. There are alternative ways to do this, and really the alternative is don't do this. Okay? Instead of breaking up the genome into many, many tiny pieces, you can think about just looking at the pieces that are the site you care about. Okay? And I keep doing this, but and many of these, there's a number of a stream of methods that really rely on the same idea, which is instead of breaking up the genome into pieces, look specifically at your binding site by tethering reagents or enzymes to that site. Now, one of the first methods for this was developed by Bas van Steensel and Steve Henikoff's group many years ago. And the idea was you could map a component of chromatin by tethering an enzyme to it. And in his case, he developed a method called DAM-ID. And what that was a chromatin protein that was made as a fusion protein to the DAM methyltransferase from bacteria. Okay? And the idea was that when that fusion protein is bound to its target site in the genome, the DAM methyltransferase will methylate the DNA here. And then you can map where your factor is, not by trying to fish out this piece of DNA, but by mapping where the methylation occurs in the genome. Okay? And it's an example of a tethered system to look to see where your factor is localized. Now that worked very well, actually. And later successive methods were similar in that similar ideas of using, instead of DAM methyltransferase, you can use a micrococcal nuclease, which will cut DNA as a fusion, as part of a fusion. And in that case, now you're not mapping methylation of the DNA, you're mapping cleavage of the DNA here. Okay? This relies on a fusion protein, and this was developed actually by Uli Lemley a number of years ago in what he called chromatin. Actually, I don't remember what the E stands for, but sorry, I should remember. But he actually, in the same paper, developed a second method, which was a little more general. Instead of making a fusion protein here, what you can do is you can tether micrococcal nuclease not as a fusion protein, but with an antibody intermediate. Okay? So now it becomes a more modular system where you can have a chromatin protein. You're basically building a sandwich at a binding site, a chromatin protein, an antibody to specifically recognize that chromatin protein, and then in Uli Lemley's case, he made a fusion protein of protein A and eminase micrococcal nuclease that would bind to the antibody. And so you're building a sandwich at the binding sites, and that is what cuts the DNA. And this gets you away from the problem of fragmenting the whole genome into pieces. Instead of fragmenting the whole genomes, now you're just cleaving at your binding site, and that vastly simplifies the problem of mapping a factor. So what I'll tell you about is an implementation that we've spent a couple years on, really, of this CHIP method, which is chromatin immuno cleavage. Okay? And we implemented it into a format that was a sequencing-based format. And the method really has a number of features which improve the efficiency and sensitivity of the method and really give you a very general method for looking at chromatin. Okay? And I'll go through some of the features. What we do is really a one-pot reaction where everything happens in one tube, and it starts really by just adding cells to that tube, and at the end you get out the binding sites, the DNA that you care about. After a few iterations, we settled on this method, but essentially what we do is we use intact, unfixed cells, and we bind them to magnetic beads. Magnetic beads are coated with concanavalin A that binds glycoproteins that are on the cell membranes. And so we can anchor our cells to these beads, and then all our successive washing steps and reagent steps are really using a magnet to capture these cells, exchange reagents, and then go on to the next step. And the steps are, we first permeabilize those cells, so there's still actually cells there, but now the reagents can come in and out of the cell and into the nucleus. And the first reagent we soak in is an antibody to a specific factor we care about, either a transcription factor or histone modification. We then wash that out, and really this is a lot like immunocytology in a lot of ways. We have an antibody now binding here, starting to make our sandwich. In the next step, we soak in a protein A eminase fusion, which now is tethered to the site of that antibody. And then we can trigger cleavage by adding calcium, which will now cut the DNA. Okay? And this is where you're now cleaving just at your binding sites. One of the advantages of this method is, as we're diffusing these reagents in at first and washing on the material, when we cleave the DNA, actually, those cleaved particles can now diffuse out. So at the end of the procedure, we have all the vast amount of DNA we don't care about still stuck on our beads. And the DNA we do care about, the sites that are the binding sites for our protein, are actually what leaks out, and we collect the supernatant and purify DNA from that, and it gives us a very clean DNA preparation where we have the sites we care about. The vast amount of the DNA is left behind. It does not go into our later reactions where we make libraries and then do paired end sequencing of these fragments to map where those binding sites are. This turns out to be very efficient. It's published. We've used it a lot. This is an example for histone modifications. This is a modification for lysine 27 trimethylation, that repressive mark I told you about, on human cell lines. And we can compare it to data, this has been looked at a lot, and we can compare it to data from ENCODE, for example, on the same cell lines and antibodies. And you can see that with ENCODE, you actually have to do quite a bit of sequencing, but you get a very good landscape across a large chromosome region here, where here's a block of genes that are coded with the histone modification and are silenced, transcriptionally inactive, other blocks which do not have the modification. And with cut and run, with this, I should have told you actually, it's cleavage under targets and release using nuclease, we get actually a very, very good picture of this genomic region from much fewer reads. And actually, if you look at it, you can start to realize actually our signal is just as good with less background, okay? Now, this is in some ways not a really fair comparison because you have a lot of reads here and fewer reads here. We can downsample the ENCODE data to really directly compare what their relative efficiencies are. And now you can see actually that with relatively few reads, we get a much better landscape than you do from a chip kind of procedure where it's very hard to distinguish signal from noise. Okay, so again, I mentioned this is published. We actually have, we were at this state really about a year ago. And what I'll tell you about in my remaining time is what we've been doing since then, really. And it really breaks into three little brief vignettes. One is, because of these efficiencies, we can really think about doing this in a very high throughput manner so that many, many proteins can be characterized or many different cell types. And I'll show you how we're doing that just in really just 2 slides. The other thing we can think about is because this works so well, we have a project in the lab that not just mapping the features of chromatin one factor at a time, but you can start to think about complexes of proteins that you can query in a cell. And then finally, I'll show you our very, very recent work on improving the method even further. Okay. So we want to look at many different cells and many different epitopes. And so the way that we've been doing this is really converting this into a fully automated robotic platform. We use a biomech liquid hand in the robot. This is the work of Derek and Jay in the lab. And really, they were thinking about different kinds of samples you can put in, either cell lines, dissected tissues. In this case, this is xenografts of a tumor, a human tumor that's grown in mice that we can then treat. And actually, it doesn't really matter what you use. We treat them all the same because the method, as I mentioned, is modular. You have cells of interest, and once you mix them with an antibody that you care about, they're all going to go through the same steps and procedures. And so for something like a tumor sample, we disaggregate the cells, we mix them with Con A beads, magnetic beads again, and then they go into a well of a 96-well plate, and everything else happens on the robot, where we go through the entire cut and run procedure of successive binding and washing steps, adapter ligation, and then PCR enrichment. And so at the end of the procedure, you end up with libraries that you can then pool and mix and run on a multiplex sequencer. Okay? The reason we're excited about this is because this really moves us out of the spectrum of characterizing cell lines for just a couple epitopes or just under a couple conditions, where we can really think about specific mutants or specific drug treatments or anything where you would want to look at many, many different parameters. And the reason why I think it works is because the procedure is efficient. We don't need very many reads. We don't need very much material. And so when we calculate how much it costs to run this robot, and we've run this robot quite a bit now, we end up estimating that it's going to cost us about $80 to characterize the chromatin profile for a cell population and an epitope, which I think is very tractable. And you can imagine setting this up as a core facility where many people could come with whatever kinds of cells they care about, and with the antibody that they care about, and that goes into a well. And at the end of the procedure, they get back sequencing data for their epitope. Right. The next thing that I'll tell you about is this idea of looking at different epitopes at the same time. So Sandepan in the lab actually had a very specific problem, and what he was interested in chromatin remodelers, which are known to move nucleosomes around. And this, I think, is a nice implementation of being able to leverage the efficiencies of the cut-and-run procedure to do new things. Okay? So this is a cryo-EM model of the RISC chromatin remodeler. It uses ATPase to push ATP to push nucleosomes around. And in this cryo-EM model, in this cryo-EM model, you have the RISC complex wrapped around this gold nucleosome here. Okay? We know from functional studies that RISC is important to maintain the open chromatin structure of promoters, of active promoters. Okay? But one controversial subject or topic in the field has been that we know that promoters are typically nucleosome depleted. I keep getting these buttons wrong. But there's been conflicting evidence about whether there may be some kind of fragile nucleosome or more unstable nucleosome particle that actually occupies promoters in some cases. Right? And one specific hypothesis that Sandopan had was that maybe the nucleosomes in these promoters, these fragile nucleosomes, are actually RISC-bound nucleosomes here. And so now you can imagine that we can test that if we were to do the following experiment. We would do a first experiment where we cleave, using cut and run, all the particles that contain RISC. And then we use that material as the input for a CHIP reaction where we ask, well, are there actually histones in that particle? So by using these two orthogonal techniques, you can ask about where is one protein and is the second protein there at the same time. And that's exactly what he did. And that procedure goes like this. It's really the same cut and run procedure for all the first steps. But at the end of that, he takes the supernatant of these chromatin particles and uses a flag peptide. His first protein is flag tagged. He uses a flag peptide to disassemble this sandwich and then uses an antibody to another chromatin protein to ask, is that second chromatin protein in the chromatin particles that he's released? Okay? This has been done before by using in a procedure called sequential CHIP, where you do a CHIP procedure and then another CHIP procedure. But because of the inefficiencies of each of those steps, those experiments are typically very messy. They're hard to interpret. They're very hard to normalize. And here, by using two different methods, we actually get a much clearer experimental procedure. So one control experiment would look like this. This is a procedure he called CUT&RUN.CHIP. It's actually, it was published this week. And where he did, in his first step, a cut and run reaction for the H2AZ variant nucleosome. You get a landscape in the yeast genome where you see sites where H2AZ is found. And then he can then, in a second step, query saying, well, of all those chromatin particles I've released, what's in them? Okay? For an IgG control, you see that there's nothing. For H2AZ itself, you see that actually there is H2AZ. That's very good. That's that we know that's there because that's what we targeted in the first step. And then you can ask, well, what else is in these particles? H2B that's an obligatory component of that nucleosome. And then specific histone modifications are there, but not other ones. Okay? So then he could do his experiment, which is to ask, well, what about these RISC particles? If I target RISC, release RISC chromatin particles, what histones are in there? What histone modifications? Or are there any? And that experiment looked like this, where he does a RISC cut and run. You get a landscape of where RISC is cleaving out chromatin particles. And then you can ask, well, what's in there? He found that H2AZ, many histones with specific modifications are in those histones. And this demonstrates that those regions of the places where RISC is bound also have histones on them. And he concluded from this that there are actually these fragile nucleosomes that had been implicated from some previous studies, but were controversial. Okay. And that was, we think actually, and we think those are dynamic particles, and the reason why they're hard to detect is because these nucleosomes are actually being pushed around and are partially unwound. Okay. So in the last few minutes, I'll tell you about a third subject, which I think is changing what our capabilities are to do, and that's why I wanted to tell you about it today. So I told you about cut and run. That was a more efficient, more sensitive method and compared to CHIP reactions here. And one thing we were interested about was can we then dial down the amount of material we need to, because if we do have an efficient, sensitive method, we should be able to use less material. And that was true. You can see that in all these cases, we're looking at about 7 million reads. But you can vary your amount of input cell culture or cell material down to quite low amounts of material where really we're profiling only 100 cells here. And so it's very sensitive. Is that the end or is that 5 more minutes? OK. Really, I have only 1 more slide or 2 more slides. So that's good. And there was some work saying that we could potentially use cut-and-run to look at individual cells, although it's really on the edge. And what we switched to was a different method that I'll very quickly tell you about, where since I told you that the method is a modular method, really, and it uses protein MNAs to bind any antibody you want, you can equally imagine that you could use a different enzyme at that step. And the enzyme we wanted to use was TN5 because it integrates adapters for sequencing instead of cleaving the DNA. And the comparison of the methods are here, where in a cut-and-run method, we bind protein A, we collect a supernatant after cleavage, and then process the material to make a sequencing library. By developing CUT&Tag, where instead we use protein A and TN5 fusion to bind to this antibody, we can then activate the enzyme, integrate adapters that make a sequencing-ready library. And that greatly speeds up the procedure. Now we're talking about essentially being done with the experiment from cells to sequencing libraries in essentially a day. Okay? And again, since it's a one-pot reaction, you can do it in an automated format. So, sorry, there's some details there. The last thing I'll tell you about is that the sensitivity of this method is actually higher than cut-and-run. The background is lower, okay? And so we can profile all the modifications that we've looked at by cut-and-run using CUT&Tag and with more sensitivity. And what this really does is allows us now to look at single cells. Okay? You can imagine your experiment would be, this is really just a review of what I told you, that we need less and less cells. But this encouraged us to say, can we actually map chromatin features in an individual cell? The way we did that was using an iCell 8 machine that Takara makes. So we could really do our reactions under ideal conditions and then aliquot them out to individual cells and ask about those individual cells. And the way we do that is diagrammed here. We take any cells we care about. We do the cut-and-tag reaction in bulk, but the material that doesn't leak between cells, so they're still in individual cells in this point. And then we aliquot the cells using this into a nanoarray of 5,000 wells using a dispenser so that each well has one cell. Okay? And at this point, we do the PCR enrichment of the libraries using barcoded adapters, barcoded PCR primers. After the PCR, we then pool it all back together and do a bulk sequencing and decode individual cells from the barcodes. Right? And it turns out this works very well, and I'll end on this one slide, where we can now profile in individual cells here as rows here for the top, I think, 600 cells of an experiment. There's probably 100 cells from an experiment. That here's a domain that we can see in bulk, and we get a very clear image of that from individual cells. So we think we can now look at active, it's the same thing, it's true for active histone modifications. We can look at individual cells for what's active and what's repressed, and that way get to this picture that I first told you about of what is the status of chromatin and gene expression in individual cells. And I'll end there and take any questions if I have any time. Thanks.
We have time for just one question.
Thank you. Do you foresee the ability to multiplex cut-and-run with other techniques such as RNA-seq in the same cells?
I think you certainly could do that. We haven't actually done any work on that. We're more interested in multiplexing for different chromatin proteins. But I think, yes, especially with strategies that use barcoded reagents, that would be particularly appealing, actually, on the single cell level.